In this video, we're going to highlight the main points of Chapter 17, which is looking at the citric acid cycle. We'll go through and look at an introduction or the purpose of the citric acid cycle, the link between glycolysis and the citric acid cycle, and the process involved there, the steps involved in the citric acid cycle. We will highlight some regulation or control points. And then we'll also look at how the citric acid cycle is a source of biosynthetic precursors. And lastly, we will focus on, briefly, the glyoxylate cycle. So we've seen the process of glycolysis, which converts glucose to pyruvate. And then we know that the pyruvate has three distinct fates. Pyruvate can undergo fermentation, whether it be lactic acid fermentation or alcohol fermentation, and those are under anaerobic conditions. But under aerobic conditions, that pyruvate is going to be converted to acetyl-CoA, and that acetyl-CoA can then enter the citric acid cycle. So the aerobic processing of glucose begins with complete oxidation of glucose derivatives to carbon dioxide. The citric acid cycle is the final common pathway for the oxidation of fuel molecules. Most fuel molecules will be converted into the acetyl-CoA so that they can enter this cycle and ultimately be converted to CO2. So this actually got cut off, but this is CO2 here. So we're going to go through and look at the steps that are involved in this overall process and the conversion of pyruvate to our acetyl-CoA. And this is just a figure illustrating here the structure of acetyl-CoA. We do simplify this structure and just indicate that we have an acetyl portion that's connected to this CoA portion that's over here. So the citric acid cycle takes place in the matrix of the mitochondria. Remember that glycolysis took place in the cytoplasm of the cell. And this is for eukaryotic cells. So we've gone from the cytoplasm now into the mitochondria, where this process is going to occur. Now the citric acid cycle serves the purpose of harvesting high energy electrons, and it does so in the form of the NADH and the FADH2. In this process, you have a 2-carbon acetyl unit from the acetyl-CoA. That's the acetyl portion of that acetyl-CoA. That comes in and condenses with oxaloacetate, and it forms citrate. And then citrate is going to be subsequently oxidized through this process. And the high energy electrons will ultimately be used to reduce oxygen to water. In the process of this reduction, we're going to generate a proton gradient, and that proton gradient is used to synthesize ATP. So the citric acid cycle is part of an overall process, and that overall process is referred to as cellular respiration. It involves the removal of high energy electrons from carbon fuels, and it does so in that form, again, of the NADH and the FADH2. And these electrons will ultimately reduce oxygen and generate that proton gradient that's used to synthesize ATP. And so we outline it here just so you can see how all of this is put together. So if we're looking at this, we have the citric acid cycle here, but then we have oxidative phosphorylation to the right of that structure. And the oxidative phosphorylation is referring to the electron transport chain and the proton gradient, and associated with this will be ATP synthase, which is talked about in future chapters. So the citric acid cycle itself is not going to generate a large amount of ATP, and we're going to see that as we go through the steps. It also is not going to include oxygen. So we couple the citric acid cycle with this process of oxidative phosphorylation. What the cycle does do, it removes electrons from the acetyl-CoA and uses these electrons in order to reduce the NAD plus and the FAD so that we can form these electron carriers here. And then we'll have the electron carriers go in towards this electron transport chain. They'll release those electrons, and that will be used to generate a proton gradient across the mitochondrial membrane. That proton gradient will flow through ATP synthase. It's going to generate ATP from ADP in that process. In this, we have ultimately electron carriers will result in about 9 molecules of ATP when they're oxidized by O2 and oxidative phosphorylation. But we'll kind of bookkeep as we go through. And again, this portion is not going to be our focus here. We're focusing on this portion. Those of you in second semester will start by looking at the oxidative phosphorylation. Okay, so pyruvate, we said, had one of 3 fates, and one of those fates was to be converted to acetyl-CoA under aerobic conditions. And so we have to have a means to, or pathway to take that pyruvate and convert it to acetyl-CoA. We have what's referred to as the pyruvate dehydrogenase complex, which is going to link that pyruvate that's generated in glycolysis to the citric acid cycle. This pyruvate dehydrogenase complex is a component of the mitochondrial matrix, and it actually is composed of 3 distinct enzymes that kind of work together. The pyruvate that's produced by glycolysis will be converted into acetyl-CoA, and then that acetyl-CoA here, so we have this process, pyruvate to acetyl-CoA, and it's here that our pyruvate dehydrogenase complex is going to come into play. That acetyl-CoA, once we've generated it, will then serve as fuel for the citric acid cycle so that we can generate those electron carriers, which would be passed along to oxidative phosphorylation. So the pyruvate dehydrogenase complex, there are, we said, 3 enzymes which are associated with the complex shown here, and we abbreviate these as E1, E2, and E3. Pyruvate dehydrogenase component is E1. This dihydrolipoyl transacetylase is E2, and the dihydrolipoyl dehydrogenase is E3. And we can see the number of chains that are associated with these, and the prosthetic group or the coenzyme that's associated with these processes. It also outlines the reaction that it catalyzes. So this is a multi-step process that's all controlled by this one complex there. Now, what we're looking at is the pyruvate dehydrogenase complex that exists in E. coli. This is a simplified version so that we can better understand what's going on. Under aerobic conditions, that pyruvate will be transported into the mitochondria by a carrier protein that's embedded in the mitochondrial membrane, and that carrier protein is part of that pyruvate dehydrogenase complex. This link, this pyruvate dehydrogenase complex, which serves as the link between glycolysis and the citric acid cycle, is an irreversible process. We have a large complex here. It's highly integrated. We have 3 distinct enzymes, and they all work together in order to allow this link between glycolysis and the citric acid cycle. So the mechanism of pyruvate dehydrogenase complex is very complex and involves a number of steps. So these steps include decarboxylation, oxidation, formation of acetyl-CoA, and then one which was cut off here when I converted the file is regeneration of oxidized lipoamide. Okay. So this is showing the overall process here, and we're going to outline these in a little bit more detail. So in the first step, that's our decarboxylation step, Now this is all not yet part of the citric acid cycle. This is how we take that pyruvate and convert it into acetyl-CoA so that it can go into the citric acid cycle. So we have the decarboxylation step, and here our pyruvate dehydrogenase E1, that first enzyme portion, will catalyze this process. Our pyruvate is going to combine with an ionized form of coenzyme, and our coenzyme is this thiamine pyrophosphate, or TPP. So TPP is converted into an ionic form here, this carbon ion of TPP, and that substance is going to interact with our pyruvate. It'll combine with pyruvate. There actually is a 4-step mechanism that's involved in this, and we'll see that in the next slide here in a second. But the TPP, which is that coenzyme, or that prosthetic group, is going to form this carbon ion, and the carbon ion is going to attack the carbonyl group of pyruvate. Okay, going to attack that carbonyl group of pyruvate, and decarboxylation will occur. The charge of that TPP carbon ion is going to help stabilize this process, and we'll be able to go through and form this intermediate here, hydroxyethyl TPP, which we'll then see goes through several subsequent steps. So our overall mechanism is here. We start with this TPP, which again is a prosthetic group that is part of that E1 portion of the overall pyruvate dehydrogenase complex. That is going to undergo deprotonation and form this carbon ion, so this TPP ion, which will then attack that carbonyl group of pyruvate. This process is going to be followed by decarboxylation of pyruvate. The ring of the TPP will help stabilize the charge. The intermediate is then going to be protonated. So there's our protonation of that intermediate. And then our hydroxyethyl TPP is going to be formed. That hydroxyethyl TPP is then going to undergo conversion so that we have a 2-carbon fragment which is oxidized and transferred to this lipoamide, and we're going to go through and ultimately form this acetyl-lipoamide here. This is now involving E2. Once that occurs and we have the formation here of the acetyl-lipoamide, E2 is going to catalyze the transfer of the acetyl group to coenzyme A. And that's going to allow for the production of acetyl-CoA. We also have the result of that transfer, which is this dihydrolipoamide. Now, this portion we're going to have to take and regenerate in order to allow for the enzyme to continue to function. And that's our next step is taking that dihydrolipoamide and regenerating that lipoamide here, which can then undergo subsequent cycles. So just looking at the structure of this pyruvate dehydrogenase complex, these 3 enzymes are very highly integrated, and it's a very complex structure. The lipoamide arm that's participating in this reaction is going to allow for movement of substrates from one active site to another within the complex. We have 3 different enzymes functioning, and so we have to be able to move things between these. Now this is that lipoamide arm, and in the previous slides, that portion we were looking at involved in the reaction is down here. So if we want to look at what's going on in this overall process, we start with the enzyme that's ready for a catalytic cycle. Now, if we're looking at step 1, the pyruvate is decarboxylated to form that hydroxyethyl TPP. That's that purpose of step 1. And then we go on to step 2, where the lipoamide arm of that E2 portion, we see the arm here, is going to move into the active site of E1, and that's going to allow for reactions to take place. In the third step, that E1 catalyzes the transfer of the 2-carbon group to the lipoamide group to form that acetyl-lipoamide complex, which we see there. In step 4, we're moving down. We have this E2 that catalyzes the transfer of the acetyl moiety to the coenzyme A, and we ultimately produce acetyl-CoA as our product. So the first 4 steps are simply to produce this acetyl-CoA, which can then enter the citric acid cycle. Once we do generate that, though, we have to have additional steps so that we can take this arm here and get it back to its starting position. So that dihydrolipoamide arm is going to swing to the active site of E3, and that E3 is going to catalyze the oxidation of that arm. And that's occurring in step 5. Okay? So it swings up into that E3, and then we're going to have that oxidation of that arm. And then there's a transfer of protons and electrons to NAD+, and that's what's happening here in step 6. And that is completing that particular reaction cycle so that we can then go through it again as more pyruvate is coming in and needs to enter the citric acid cycle. So here we're looking at the overall citric acid cycle. Acetyl-CoA is the fuel for the citric acid cycle. It begins with the condensation of a 4-carbon unit and a 2-carbon unit. So the 4-carbon unit is the oxaloacetate, and the 2-carbon is the acetyl group of acetyl-CoA. This will produce coenzyme A and citrate through an aldol condensation reaction, and the reaction is followed by hydrolysis, which will be catalyzed by citrate synthase. Now, in this overall process, the side reactions must be minimized. And the way that we minimize it is through the mechanisms of the different enzymes that are present. Some of these mechanisms we're just going to not go into too much detail simply based on time. But we will talk about this general cycle and the individual steps that are involved in this cycle. Again, we won't so much focus on the mechanisms that are involved in controlling each of these enzymes, though. So we have the oxaloacetate, which is going to interact with that acetyl portion of the acetyl-CoA, and we produce citrate here. Okay. So if we begin by looking at citrate, with this, our hydroxyl group is not properly positioned. So citrate is going to be isomerized, and it's going to be isomerized to form this isocitrate, which we see here. This is going to be done via a dehydration step, followed by a hydration step, the sum of which is shown in this figure, but the actual reaction that's taking place is our citrate to this cis-aconitase, and then finally to isocitrate. So this is going to be a dehydration step. We're removing water in this step. And here we have a hydration step where we're adding water back to it, just in a different position. So this process is what represents the conversion of citrate to isocitrate via this enzyme aconitase here. After this step occurs, we have the first of 4 oxidation reduction reactions. So the isocitrate will be oxidatively decarboxylated, and it's going to be done so using the enzyme isocitrate dehydrogenase, and that's going to form an intermediate, which is called oxalosuccinate. This is an unstable substance, and so it's going to be almost immediately converted to this alpha-ketoglutarate. This is a step where we see our first loss of CO2. So we've taken one of these carbons and broken it down to CO2. We also generate the first high transfer potential electron carrier of the cycle in this step. So this step is broken down into isocitrate to oxalosuccinate. And again, that oxalosuccinate is an unstable intermediate. This portion here is where we're taking that NAD plus and producing our first NADH of the cycle. That oxalosuccinate is going to then be converted to this alpha-ketoglutarate, and it's this step where we generate the first CO2. But these reactions together make up this conversion of isocitrate to the alpha-ketoglutarate, and it's done so by using this isocitrate dehydrogenase enzyme. This process is going to be followed by a second oxidative decarboxylation reaction, where we're going to take that alpha-ketoglutarate and form succinyl-CoA. This particular reaction is going to be catalyzed by alpha-ketoglutarate dehydrogenase complex. And this particular complex will function in a way that's homologous to the way that pyruvate dehydrogenase complex works. So there are similarities between the alpha-ketoglutarate dehydrogenase complex and the pyruvate dehydrogenase complex. Once that occurs, this succinyl-CoA, there's a thioester bond that's present there that is going to be cleaved. And the cleavage of that thioester bond is coupled to the phosphorylation of a purine nucleoside diphosphate, usually ATP. This step, the succinyl-CoA to succinate, is the only step in the citric acid cycle where we're generating ATP. It's the only step that directly yields ATP or an equivalent. And here, the resulting product is going to be our succinate. And we're also going to have CoA being released and ATP. That succinate is then going to go through a multi-step process by which the sole purpose is to regenerate this oxaloacetate. So succinate is going to be oxidized to form fumarate, and it's done so with the enzyme succinate dehydrogenase. We'll see this is the step where we produce another one of those electron carriers, FADH2. The fumarate is then going to undergo hydration to form malate. So we have water being added to form malate. The malate will then be oxidized using malate dehydrogenase, and will produce or regenerate that oxaloacetate, and will also have another one of those electron carriers present. So this is the overall citric acid cycle, and you want to be familiar with the reactions that take place along this cycle. You also want to be familiar with those two steps where I've outlined additional reactions that take place. So both here and here. Here we're looking at a table which summarizes the reactions that we looked at in the previous slide. The formation of acetyl-CoA from pyruvate. Remember, again, this is irreversible in animals. The acetyl-CoA that is generated has two principal fates. It can go through metabolism by the citric acid cycle, or it can actually go to incorporate into fatty acids, which we'll talk about later. But if you look at these overall, let's just highlight the net reaction that's taking place for the citric acid cycle. So acetyl-CoA plus 3 NAD plus FAD plus ADP plus the inorganic phosphate plus 2 water. These are the reactants, net reactants. And our products carbon dioxide, 2 of those, 3 NADH, 1 FADH2. Those are our electron carriers. 1 ATP equivalent, 2 protons, and coenzyme A. So overall, 2 carbon atoms will enter the cycle. Those will leave in the form of CO2. 4 pairs of hydrogen atoms will leave the cycle via the 4 oxidation reactions, and those are going to leave in the form of the NADH and the FADH2. And then we have 1 ATP equivalent being generated and 2 water molecules being consumed in this process. So we mentioned that the conversion of pyruvate to acetyl-CoA is irreversible, and therefore that acetyl-CoA cannot be converted back to glucose. So that acetyl-CoA, we said, had 2 fates. It can be oxidized to CO2 by the citric acid cycle, or it can be converted into lipids. Now the fate of the acetyl-CoA is going to be dependent upon, so there's our 2 fates here, it's dependent upon the control of that pyruvate dehydrogenase complex. Now the control or the regulation of that pyruvate dehydrogenase complex is going to be through this kind of on-off switch based on phosphorylation of the complex. So the complex can be phosphorylated by pyruvate dehydrogenase kinase. And if that complex is phosphorylated, then it switches it off or inactivates it. But then when we need that to be on, you can reverse the process by another enzyme, our pyruvate dehydrogenase phosphatase. That is going to then activate that complex and allow for substances to continue through that citric acid cycle. The kinase and the phosphatase are going to be very highly regulated enzymes. And we can see some of this regulation here in this figure. So we've seen that when we have high energy, the complex is inhibited by its immediate products. So here is our high energy. And this pyruvate dehydrogenase complex, which is allowing the conversion from pyruvate to acetyl-CoA, if we have high energy, the products of this, acetyl-CoA, the NADH, and even ATP will inhibit the function of this enzyme. So it turns it off. Now when we have low energy, we have activation of this complex. So pyruvate is going to serve as an activator of the complex. The ADP is also going to serve as an activator of the complex. So this will inhibit the kinase that phosphorylates the pyruvate dehydrogenase complex. So the complex remains turned on. There are control points of the citric acid cycle, just like any other cycle. We've seen that we can regulate this pyruvate dehydrogenase complex here in terms of converting that pyruvate to acetyl-CoA or taking that and using it for fatty acids. But then we have 2 additional control points along the citric acid cycle. The first is this portion where we have the isocitrate dehydrogenase enzyme in there. And this particular enzyme is going to be allosterically stimulated by ADP, so it's turned on by ADP, but it's going to be inhibited by ATP and NADH. Then the second control point of the citric acid cycle is going to be at this alpha-ketoglutarate dehydrogenase enzyme step, where here we see that ATP, succinyl-CoA, and NADH will all inhibit this enzyme from functioning. So when the cell has enough ATP, the rate of the citric acid cycle is going to be reduced. We don't want to use the cycle if we don't need to. Now, the citric acid cycle can also serve to provide intermediates for biosynthesis. Many of the amino acids are going to be derived from some of the substances here. The individual reactions that take place are looked at in other chapters of the text, but here what we want to look at is you have your pyruvate being converted to acetyl-CoA and entering this cycle. Now that citrate can undergo some subsequent reactions to produce fatty acids and sterols. If we get down to alpha-ketoglutarate, we see that the alpha-ketoglutarate can be converted to amino acids. The succinyl-CoA can be converted to some porphyrins, heme, chlorophyll, some other substances here. And oxaloacetate can go on to form some amino acids. So there are lots of side reactions that can take place, or that the intermediates can serve as precursors for other things that are needed. Now, the intermediates of the citric acid cycle, because they're often drawn off for various biosyntheses, we need to be able to replenish these. And replenishing of these intermediates is done through a variety of different processes, some of which we've already looked at. So glycolysis can help to maintain a flow of the intermediates here. The citric acid cycle can continue to regenerate substances that will continue to create these intermediates. We also have oxidative phosphorylation and fatty acid oxidation, which will come in and help regenerate these intermediates so we can continue this process. So up until this point, we've looked at the citric acid cycle, the steps of the citric acid cycle, the regulation of the citric acid cycle, and how the intermediates can be used for other substances, and the need for regenerating those. Now here in this slide, we're looking at the glyoxylate cycle, which is a different cycle. This pathway is going to allow the conversion of acetyl-CoA generated from fat stores into glucose. And this is going to be present in plants and some microorganisms. This process is similar to the citric acid cycle, but the difference is in the glyoxylate cycle, there are 2 decarboxylation steps which are bypassed, and 2 molecules of acetyl-CoA enter per turn of the cycle here. Okay, 2 per turn, compared to the 1 acetyl-CoA that enters per turn of the citric acid cycle. So we can see here 2 acetyl-CoA going in, whereas in the citric acid cycle it was only 1 acetyl-CoA around. We can look at the net reaction of this process. Our reactants are 2 acetyl-CoA plus NAD plus 2 H2O. And our products, succinate, 2 CoA, NADH, NH plus. Okay, so this is the net reaction for the glyoxylate cycle. These reactions will take place in glyoxysomes of plants, and the succinate that's generated here could then be converted into carbohydrates that could fuel the plant. So that's just a brief overview of the glyoxylate cycle. Again, there are really 2 differences here. The first is that we have 2 decarboxylation steps which are bypassed, and then the second is that we have 2 acetyl-CoA per turn versus the 1 in the citric acid cycle.